Hi everyone, I'm Sharon from Philip Morris International Research Laboratories, Singapore. Today, I'll be presenting on the comparative assessment of pharmacokinetics and acute lung inflammation of nicotine dry powder aerosols generated by PreciseInhale. To start off, you might be asking, why are we delivering nicotine in a dry powder? One of the reasons is that nicotine has a complex dosimetry when inhaled as a vapor aerosol mixture. Hence, our group hypothesizes that harnessing nicotine in a dry powder aerosol may allow better control of its delivery to the lungs. A previous study by our group has shown that a batch of nicotine-containing dry powder, batch A, can be successfully aerosolized and delivered intratracheally to rats via the PreciseInhale system. In this study, batch B was produced for comparison to batch A. I will elaborate more on the objectives of comparison in the following slide. Briefly, the powders were produced via spray drying and contained approximately 2% nicotine and other excipients. Moving on to the objectives of this study, we aimed to address two main research questions. Firstly, how will the aerosol characteristics and nicotine pharmacokinetic (PK) profiles of both batches compare? Secondly, will these batches when inhaled cause acute lung inflammation in rats? To address these research questions, I will briefly run through the methodology adopted. Firstly, the aerosol generation and exposure system used was the PreciseInhale from Inhalation Sciences. It is labeled as PI in this schematic. To give you a brief background, PreciseInhale is a single rodent exposure system. It is capable of achieving precise bolus dosing through its real-time monitoring of aerosol concentration as well as the connected rat's breathing pattern. In our study, the rat was first fully anesthetized and then intratracheally intubated with a catheter. The intubated rat was then connected to the PreciseInhale. Upon the start of exposure, high-pressure air was released into the powder chamber to aerosolize the loaded powder into an aerosol cloud. This was then breathed in by the connected rat. The target dose to be achieved in our study was 0.1 milligrams nicotine per kilograms body weight. The corresponding powder dose was input into the PreciseInhale software. Once the target dose was reached, the exposure was automatically terminated by the software. Next, one of the key aerosol characteristics to be assessed was the aerosol's particle size distribution, or PSD for short. In this study, the aerosol's PSD was determined using a MARPO cascade impactor. The cascade impactor was connected to the exposure module of the PreciseInhale, as shown in the image on the right. Upon exposure, aerosol was collected on each stage of the cascade impactor, and the amount of aerosol deposited per stage was determined gravimetrically. From the PSD data, the mass median aerodynamic diameter, MMAD, and geometric standard deviation, GSD, of the aerosols were then derived accordingly. For PK analysis, blood was sampled at various time points up to 4 hours after the start of exposure. The blood sampling regime is as shown below. Blood was sampled via a tail vein catheter inserted into the rat's tail, as shown in the image on the right. Nicotine and cotinine concentrations were then quantified in blood plasma, and PK profiles were derived. Lastly, for the lung inflammation analysis, the exposed rats were terminally dissected either at the 6 hours or 24 hours post-exposure time points. The bronchoalveolar lavage fluid, or BALF for short, was collected at these terminal time points. Briefly, PBS, or PBS-BSA, was used to flush the lungs of the rat to collect BALF. BALF was then spun down, and the supernatant of the first lavage cycle was used for Luminex measurements of pro-inflammatory cytokines. On the other hand, the pellets from the first to fifth lavage cycles were used for differential cell counting via flow cytometry. Now, let me move on to the results of this study. Firstly, dry powder aerosols generated from both batches were shown to have similar MMAD and GSD of approximately 4 microns and 1.8 respectively. From the PSD data, the aerosols were thereafter determined to be inhalable using the multiple path particle dosimetry software, as the weighted theoretical lung deposition percentages of A and B were 58 and 56% respectively. Next, the target dose was also delivered within short exposure durations of less than 5 minutes from both batches. variabilities in exposure duration were likely due to variabilities in the rat's breathing rates. Now, let me move on to discuss the PK data. Firstly, the nicotine PK profiles were shown to be comparable between both batches. It is also shown here that nicotine was absorbed quickly into the blood, with Tmax occurring at the first blood draw time point. Secondly, the PK profiles of cotinine, which is a metabolite of nicotine, were also shown to be comparable between both batches. Cotinine was seen to accumulate in the blood over the first 4 hours post-exposure, hence verifying that nicotine had been absorbed systemically and was metabolized over time by the exposed rats. Lastly, I will move on to the lung inflammation analysis results. Overall, there was no significant increase in the total lung cell count for both batches compared to the air control group. This indicates that no broad inflammation was observed. From this, the percentage cell count for 4 different immune cell groups were determined. It is shown here that there was no significant increase in the percentage cell count for each immune cell group, comparing either batches A or B to the air control group. To supplement this data, the pro-inflammatory cytokine measurements by Luminex also showed that across the board, both batches A and B showed little to no significant increase in pro-inflammatory cytokine expression compared to the air control group. Hence, to conclude, firstly, dry powder aerosols can be effectively generated by the PreciseInhale for delivery of nicotine into the rat systemic circulation via inhalation. Secondly, both batches of nicotine-containing dry powder are similar in terms of aerosol characteristics and nicotine and cotinine PK profiles. Lastly, dry powder aerosols generated from both batches did not cause acute lung inflammation in rats up to 24 hours post-exposure. I would like to thank the following people for their help and guidance with this project. To end off, if you have any questions regarding this presentation, or if you would like to understand more about the aerosol generation and in vivo exposure capabilities offered by us, we'll be pleased to take your queries at this contact. Thank you.